So we just finished talking about organelles and cytoskeletal filaments. So organelles, as a review, helped a eukaryotic cell organize chemical reactions and grow in size. And cytoskeletal filaments help give the cell structure and shape and are important for a variety of cellular functions. And so today we're going to think more about how proteins move around in the cell. So we're going to link those two concepts together. And I'm going to start by showing this diagram that I showed you on day 1, which is a picture of the central dogma. And in this diagram, we have DNA being transcribed into mRNA, and this actually happens in the nucleus. And then that mRNA leaves the nucleus and goes into the cytosol, where it's translated into protein. And I am showing you that this process is taking place in the cytosol. However, that's a little bit misleading. So some proteins are in fact made in the cytosol, and these include proteins like cytoskeletal filaments actin and tubulin, and enzymes that are used in glycolysis. However, other proteins need to reach different destinations. And so we've talked about some examples in class. I'm showing you 2 here. So salivary amylase, this is an enzyme that breaks down glycosidic linkages in starch. And this needs to be secreted from our cells and is present in saliva, where it actually interacts with starch. And another example that we've talked about are chaperone proteins. These proteins help other proteins fold, and they need to be in the ER to do this. But this presents a problem when we think about where these proteins need to end up. And that's because organelles are enclosed in lipid bilayers. And so lipid bilayers, as we've talked about, they have hydrophobic stretches because of those fatty acid tails. And proteins, in their 3-dimensional conformation, they're largely hydrophilic on their outward-facing side. So how do proteins then get through organelles into their final destination or get through the plasma membrane? And we're going to talk about some examples today where proteins are made in the cytosol and then trafficked into an organelle, so that's the default location, or how some proteins are actually going to be made in the ER, so adding a little bit more complexity to this picture I'm showing you here. To do this, we need 3 things, okay? We need a sorting signal, we need carrier proteins that recognize that signal, and protein channels, okay? So sorting signals are also referred to as molecular zip codes, and these direct proteins to the correct subcellular localization, just like a zip code would direct a letter to the right city. These zip codes are recognized by carrier proteins that are present in the cytosol. So these carrier proteins function a lot like mail carriers, getting those letters to the right place. Once those carrier proteins have arrived at the right place, they pass those proteins off to protein channels that are embedded in the organelle membranes, just like a mail carrier might slip a letter through a mail slot. So these are some analogies to help you think about this and keep the different molecules we talk about straight in your head. So proteins can end up in a variety of different places. So they can be imported into the chloroplast, mitochondria, peroxisome. But we're just going to focus on 2 examples today. We're going to focus on proteins that are made in the cytosol and get to the nucleus, and we'll talk about that in the next video lecture. And then proteins that are made in the ER and then move through different organelles that are called the endomembrane system. And that's what the rest of this video is going to be about. So the endomembrane system involves the Golgi, the ER, the lysosome, and the plasma membrane. And early evidence looking at cells that were specializing in secreting protein showed that these cells that do a lot of secretion, they have a lot of endoplasmic reticulum and a lot of Golgi apparatus, like a lot of these membranes. And so this suggested that these were maybe involved some way in protein secretion. And so eventually, George Palliday did these really elegant pulse chase experiments, and he worked out the order of this pathway. So if a protein is destined for secretion, It needs to get made in the ER, then it moves to the Golgi, and then it moves to the plasma membrane. Later work also showed that proteins that traffic to the lysosome also move through this system. So they exit the Golgi and are diverted to the lysosome, and we'll talk about how that happens. And some proteins, right, secreted proteins will fuse with the plasma membrane in this process called exocytosis, and they're released, but other proteins may stay embedded. Okay, so if a protein is destined for any of these locations, the ER, the Golgi, the lysosome, or the plasma membrane, they actually need to be made first at the ER. Okay, and we're going to talk about the evidence for the order of this pathway, and then how proteins get into the ER, and then how they're moved out either to the Golgi or the lysosome or the plasma membrane. So that's what we're going to talk about in this video. So first, let's talk about the pulse chase experiments done by George Paladay. And so in these experiments, he used pancreatic cells. And this was a pretty wise choice because these cells make large amounts of secreted digestive enzymes. So they had lots of ER, lots of Golgi, and they were constantly making protein. And so he did this pulse chase experiment. I'm going to explain a little bit about how this works. So to do a pulse chase experiment, you take a population of cells and you expose those cells to a high concentration of radioactive amino acids. In this case, he used the amino acid leucine. And so I'm going to just diagram out what happens. So as a ribosome is making protein, and it forms a peptide bond with leucine, okay, that leucine is going to be radioactive. And when we use radioactivity, we can actually follow this radioactive protein. It's tagged, and so we can follow it using a variety of techniques, including microscopy. The pulse period is then followed by this chase period. And so in the chase period, that radio label is washed out and you add an excess of cold amino acids. So now any newly formed proteins that are being made, we have amino acids. And for example, this one, let's say this is a leucine. So here, this is not going to be radio labeled. And so essentially this protein that's made after this pulse period will basically be invisible. And so this will allow us to look at a population of proteins that were made during a specific interval of time and to follow those proteins in subsequent series of experiments, so over more time. And so that's what Paladay did. And so here are the results of his experiments. So at 0 minutes, so right when the chase was added, if Paladay looked at his cells by using transmission electron microscopy so he could really see the organelles, he saw that most of the protein was in the endoplasmic reticulum. So right away it was in specifically the rough endoplasmic reticulum. So there were a lot of ribosomes on the endoplasmic reticulum. Then at 37 minutes, he found that these proteins had moved to the Golgi apparatus. So they were going from the rough endoplasmic reticulum to the Golgi, and he actually saw, right, this was a very high-powered microscope, he saw that the proteins were moving in vesicles. And then at 117 minutes, he saw that a lot of the proteins had left the Golgi and were now in these secretory vesicles or had been secreted outside of the cell. So this gave us this pathway here where it goes from the ER to the Golgi to the plasma membrane, and all of the passing off from the different organelles, the protein stays within a vesicle. So once it gets into the plasma membrane, it doesn't have to cross across a lipid bilayer again. Okay, so then that got people wondering, okay, well, how does it get into the ER? And so on the basis of these data and this big question, Blobel and colleagues proposed that a signal directs ribosomes to the surface of the ER. And they eventually showed that the signal was a 20 amino acid sequence that they named very aptly the ER signal sequence. So this is going to signal to the ER. and it marked the ribosome for transport to the ER. So if you were ever wondering why the rough ER is the way it is, why are ribosomes on this endoplasmic reticulum, it's because of this signal sequence that's present in proteins. Okay, so let's look at how this pathway works. And we're going to look at this in molecular detail. Okay, but before we do that, I want to introduce you to some key players. Okay, so first up, we have our ribosomes and our mRNA. And so ribosomes bind to mRNA, and as they read the sequence of mRNA, and we'll talk about how this happens, it pulls different amino acids, and it's going to catalyze peptide bonds between those amino acids. And so we have those amino acids being made. This is our, for example, our N-terminus. And there's going to be this short sequence of amino acids that comes out first. Okay. The other protein I want to introduce is this signal recognition peptide, which I'll refer to as SRP. But the name is, again, very apt because it's the signal recognition part, right? So it's going to recognize this signal, this ER signal sequence. The next one is its friend here, which is the signal recognition peptide receptor, or SRPR. And then last but not least is the translocon. Okay, now I'm going to go through how these all work in a cell, but I do strongly recommend that you sketch out this pathway. Okay, so before I do that, I just want to point out again our analogies. So this short initial sequence will be our zip code. The signal recognition peptide, or SRP, will be our mail carrier. And the translocon will be our mail slot, where the protein is fed through the membrane. Okay, so let's dive in. So I'm going to begin by showing you a schematic of the ER. So here's the ER, and this is the lipid membrane of the ER. It's a bilayer, and it separates the cytosol from the ER lumen. And embedded in the ER is the SRP receptor, and this is actually a protein complex, as well as the translocon. Okay, so these already exist in the organelle, in the membrane of the organelle. Now, in a cell, we have mRNA, and it binds to a ribosome, and that ribosome starts making protein. If that protein does not contain a sorting signal, then that protein will just continue to get made in the cytosol, and it can be trafficked to different locations like the nucleus, or it can remain right where it is. But if that sorting signal is the ER sorting signal, then SRP will take over. So SRP, it's actually a protein complex, and it will bind to this ER signal sequence, which is that short sequence of 20 amino acids, and it will bind to the ribosome. So it binds to these two separate entities. And after it binds, it carries the ribosome with this ER signal sequence to the ER. And it does this by attaching to the signal recognition peptide receptor, or SRPR. And so that's how the rough endoplasmic reticulum comes to be. It's because of SRP. Once it's bound to SRP receptor, it locks into position and it releases the ribosome and the oligopeptide. And when it releases it, it transfers it to the translocon, which is the mail slot. It's a channel protein. And then once it releases the signal sequence, it then diffuses back into the cytosol. Now, there's a few more things that happen here, like energy is involved. So this is just a simplified version of what happens, but it has the major events. Now, once SRP lets go of this signal sequence, protein synthesis can resume again. And so protein synthesis starts resuming, and this protein is fed as it's being synthesized through this translocon, and then it's eventually released. It's released into the ER lumen, and that signal sequence is chopped off, and then the protein folds into its 3D conformation. And so that's all shown again here in your textbook. And so we have the ER signal sequence, binding of SRP. SRP binds to the SRP receptor in the ER membrane. There's this transfer then to the translocon. The protein enters the translocon. It continues to be synthesized. The ER signal sequence is removed, and the protein folds into its 3-dimensional conformation. So now we have a protein that's entered the ER, and what will happen is it will fold into its 3D conformation, and it will be modified. And so starting in the ER, the protein will have sugars added to it, and this is called glycosylation, and the protein will become a glycoprotein. And it will also exit the ER, and it's going to exit the ER just like George Paladay saw in these vesicles, and it's going to fuse with the Golgi. Okay, the area that it enters is called the cis face of the Golgi apparatus. So the cis face is the face that faces the ER, and the protein will move through the Golgi apparatus and continue to gain additional modifications. So this protein is gaining more, we call these post-translational modifications, and then it ends up needing to go, after it leaves the Golgi, it can go to several locations. Okay. Some of the locations involve going back the way it came. So sometimes some proteins will return back to the cis face of the Golgi, or like chaperone proteins need to go back to the endoplasmic reticulum. And these require additional sorting signals. The default is essentially for proteins to go to the plasma membrane. If this is a soluble protein that was in the lumen, then this protein will be secreted by exocytosis. But sometimes proteins are embedded into the membrane here, and so they leave these vesicles in the membrane, and then they stay embedded in the plasma membrane. So this is how we get integral membrane proteins. So just depending on if the protein is in the lumen or in the membrane, when it reaches the plasma membrane, it'll either get secreted or it'll become an integral membrane protein. The last example that we'll talk about is that sometimes some proteins end up, after they leave the Golgi, going to the lysosome. So each protein that comes out of the Golgi and is destined for the lysosome has to have one more zip code. So this one has 2 zip codes. And this extra zip code, so it has the ER signal sequence, but it has another zip code that it gains in the Golgi. And what this is mannose 6-phosphate. So this is a few sugars and a phosphate group that are added to a protein. And this sorting signal is recognized usually by a receptor that's in the vesicles that are exiting the Golgi. And that receptor essentially directs this vesicle to the lysosome. Remember that the lysosome is an acidic organelle, and so it has a pump on the surface that's pumping through protons and making it acidic. And once this vesicle fuses, it empties its cargo into the lumen, and now we have a protein that ended up in the lysosome. And these proteins that are sorted there, they include things like acidic hydrolases that then help this organelle digest material that comes in. We'll talk a little bit about that in the 3rd video. So in summary, proteins destined for the ER, Golgi, lysosome, plasma membrane, or extracellular space need to be made in the ER. ER import requires a ER signal sequence which is the zip code, a mail carrier, which is the SRP. It requires energy, which we did not talk about, but I think it's important to acknowledge, and then the mail slot, which is the translocon. It also requires the SRP receptor. So again, kind of the mail carrier might have a handoff in this analogy. And then proteins that need to reach additional spaces, like the lysosome, need to have additional zip codes. So other ways to sort them off so that they don't go to the plasma membrane. So that's it for the video, and on to the next.